So Dr. Laura Wilson is now the current executive director of Marine Resources Council. She recently moved to the Lagooniverse from Fort Hays University, where she was a professor in the Department of Geosciences and was the chief curator at the Sternberg Museum of Natural History. And so I'd like to welcome Dr. Laura Wilson after I give my quick little updates. Sorry about that. We do have some upcoming events happening at MRC. So this Friday, July 12th, from 9:30 to 11:30, we have a mangrove workshop happening here. That is mangrove nursery maintenance. And so we'll be planting trees and enjoying ourselves in the sun until it gets too hot. And then we have an Indian River Lagoon inspirational hour just down the road at Florida Institute of Technology on Wednesday, July 24th from 8 to 9 a.m. That kicks off our action assembly for the year. And then last thing we have is first Friday Litter Patrol on Friday, August 2nd from 8:30 a.m. to 10 a.m. And you would RSVP to Ken Parks. Yes, Cara.
The owl's not lit, should it be?
It should be on. Okay. I will double check. Okay. But we'll welcome Dr. Laura.
Okay. Double check, shall we? And the speaker is.
It's because we're muted.
Gotcha. Oh, we weren't muted. There we go.
Do we dim lights at all?
Sure.
Okay.
His eyes are on, you're good.
Oh, I do the owl eyes, they're so cute.
There we go.
Okay, we still good online?
Yeah.
Okay, perfect.
Alright.
So sorry if people are disappointed that you're not getting a large animal vet to talk about charismatic megafauna, but I actually will talk about some charismatic megafauna of both past and present. So as Mara introduced, I am recently transplanted into Florida to be the executive director here, and I do come from a traditional academic setting. So I was a professor and curator of a natural history museum for 12 years. And my background in research is actually in paleontology, specifically vertebrate paleontology. One of the things that makes me well adapted to this ecosystem is actually come from a standpoint of studying ancient marine ecosystems. And the ecosystem that I've been studying for, there we go, about 15 to 20 years is a 85 million year old ecosystem. So this is what the world looked like 85 million years ago. So you can kind of see where the continents are. This is Pangea still breaking up, our last supercontinent. Sea level was really high. There were no ice caps. There were things other than warm climate going on for why sea level was so high. But you can see a lot of flooded continents. So it's a very unique world that we don't really have a whole lot of analogs for today. Specifically, I look at this big seaway right here in the middle of North America called the,
I'm sorry. Okay, I can forget you.
okay, called the Western Interior Seaway. So between 66 and 100 million years ago, North America was actually divided in two or actually three pieces by an inland sea. So these are shallow, probably only like only a couple hundred feet deep. There were no Rocky Mountains at that time. So the shoreline would have gone from pretty much kind of like where New Mexico is, or New Mexico is, and Utah over to kind of the middle of the Midwest. And so we'd have these shoreline changes as sea level changes. And this is the ecosystem that I've been studying for a while now. And because we're talking before the extinction of the dinosaurs, so the big dinosaur extinction was 66 million years ago, and these were marine ecosystems leading up to that extinction, we kind of had a different cast of characters living at that time. We hadn't seen the evolution of big marine mammals like our whales and dolphins that we have today, or our pinnipeds like seals and sea lions, filling a lot of those apex predator roles. We had big marine reptiles, so things like mosasaurs and plesiosaurs. Before then, we had ichthyosaurs. They're very dolphin-like, but reptiles, air-breathing, and a lot of questions that we have how their metabolism worked compared to our traditional warm-blooded mammals that we think about today. So this is kind of a sense of the cast of characters. My favorite term that I've ever read about this ecosystem is calling it Hell's Aquarium. This is actually my favorite piece of paleo art that I've ever seen on the Western Interior Seaway by the amazing Brian Eng, in a period called Savage, or in a work called Savage Ancient Seas. And it really kind of brings this Hell's Aquarium to life, that we have these really big predators. We have these big marine reptiles that are just teeth and actually have snake-like hinges in their jaws because they're very closely related to snakes, and have extra sets of teeth on the top of their mouth because they're squamate lizards. So think about things like monitor lizards, very closely related to that. We have huge toothed fish. So this guy is getting chomped, but is actually about a 15-foot long fish, built very much like a tarpon with the upturned mouth. And you have seabirds and pterosaurs, as well as large sharks, all within these big ecosystems. The animals that I tend to work on are the ones that you're always being shown like getting eaten by something bigger. So I did my dissertation actually on these seabirds called Hesperornis. They were really cool. They're goofy. They're flightless, so kind of ecologically equivalent to penguin, but actually swam more like a cormorant with its feet, but they still had teeth, so kind of dinosaur-like. But what I'm going to be talking about today are sea turtles. So this was some of the most recent research that I've done, and actually what kind of spurred me into the idea of I'm ready to move on and do something more conservation focused. So this kind of pushed me into how can we leverage Earth's past to understand the present and preserve the future. So I can thank a lot of my transition into Marine Resources Council from studying our charismatic sea turtle megafauna. So the sea turtles that I worked on this, the genus, so the main group of sea turtles is called Protostega. It is the second largest sea turtle of all time, second only to Archelon, which is kind of like its sister evolutionarily. So just kind of like one step, we probably had Protostega over time evolving into these larger Archelon. But these things, 3.5, I mean, these are sea turtles that are 10 to 12 feet long. So you can see that they're an additional 3 or more feet, 3.5 or 4 feet longer than our leatherbacks, which are our largest sea turtles alive today. So very, very large. And then they lived within this Western Interior Seaway between 80 and 85 million years ago. So this is the distribution that we understand them now. So they'd actually get up, probably actually up to Jersey shores, and then all the way down into Florida. It was up here at this point. It was covered by an ocean, but in kind of this Mississippi embayment, and then up this Western Interior Seaway.
Did not want to go again. It does want to go again. Okay.
Okay. So most of the research on Protostega has been things like describing new species, or like, hey, we found a fossil of them here, let's make a record of that and moving on. And there really hasn't been any work of the sea turtles in this region or in that region in terms of how they were living their life and what their ecology was. So when I talk about things like history, I'm talking about the timing of a lot of those major life events. So when you reach full body size, when you reach sexual maturity and can start reproducing, how big you grow, how fast you grow, metabolism and things like that. These are all things that are hard to understand in fossil animals and really hadn't been studied in fossil sea turtles. So the purpose of my study was to look at how Protostega grew through ontogeny. And ontogeny is just a fancy word to look at how something grows from birth to death, so from young to old. And what this can tell us about things like growth rates, how fast were they growing? Metabolism, how does that relate to kind of an endothermic, warm-blooded, ectothermic, cold-blooded lifestyle? The timing of sexual maturity, so when they could start reproducing, and then the timing of skeletal maturity, so when they reach full body size. And these are very important for us when we're thinking about ecosystems and ecosystem dynamics, especially in the world of conservation, where we want to understand the reproducing population, how long it takes individuals to get to reproductive maturity, and then how long they're producing offspring. So the technique that I use, or one of the techniques that I use is called osteohistology. Osteo means bone and histology is the study of tissue. So bone is living tissue. It grows, it repairs itself throughout our lifetime. And so to do this, I actually take bones and I cut them up. So I'll cut them, put them in a saw, embed the little chunks that I cut out in resin. So this what's being shown here is I have a chunk of bone in a plastic resin that I'm running through a diamond bladed saw to cut off little wafers. I glue those wafers just to glass slides, and then I grind them down really thin so that light shines through, and then I look at them under microscope. And what's really, really cool is that fossil bone looks just like modern bone when you put it under a microscope. So things like the vascular canals, the tubes that bring blood and nutrients to the bones, the areas that house the cells that actually lay down bone, all those actually get preserved in the fossil record. So we can do a lot with this technique for understanding how animals live. And there are four main factors that we look at when we cut open these bones and look at them under a microscope. What we look for is signs of phylogeny. So like the evolutionary history of an organism. Fish bone looks different than reptile bone. Reptile bone looks different than mammal bone because they're all different types of organisms. Ontogeny, a juvenile animal grows differently than an adult animal. Usually juveniles are growing quickly and adults are growing slowly or not at all. Biomechanics, so the leg of a swimming animal is going to look different than the leg of a running animal. And then I would argue the one that we understand the least amount is environment. And this is the one that I've always been the most interested in, is how can things like climate or even behavioral things like migration leave marks in these bones so that we can better interpret how animals were living their lives. So looking at these bone microstructure patterns, we can actually get at things like growth rates. In this case, we can look at relative growth rates. If you think about bone, bone is kind of the analogy that I use is like it's reinforced concrete. So if you've seen reinforced concrete get laid down, you're pouring concrete into a rebar framework. And that's going to give it strength and flexibility. So like if you see a road getting poured. Bone is like that too. Our bone is made up of an inorganic crystalline called hydroxyapatite. So it's phosphate apatite or phosphate and calcium. And then it also has an organic protein called collagen. And these work together to give our bones strength and rigidity while still being flexible. For most of us, we don't break a bone every time we fall down or slam into a door handle or something like that. And that's because we have both the inorganic and organic materials. And we can actually see that framework of those proteins in both modern and fossil bones. And that's what you're looking at here. You're kind of looking at how light is playing off the organization of the protein fibers. And so if you look at something like here on the left, you see these very linear arrangements, or in this case, circumferential arrangements of light, dark, light, dark. And that's showing us that this is very well organized bone. So I don't know if you're like me, but in my life, the busier and faster things go, the less organized I get. Versus if things are kind of, I have a chance to take a breath, I get a little bit more organized. Bone's the same way. When it's growing slowly, it can be more organized. Versus if we look at our dimetrodon over here, it is a very fast growing bone where it is less organized. And that's why you kind of have these bright patches going at every which way. Confucius ornis in the middle, one of our early birds, is kind of the Goldilocks in between. It's an intermediate bone. And so these are all, the alligator on the left is modern, Confucius ornis and dimetrodon are both fossil bones. And so we can see these in modern and kind of get the ground truthing and then apply that to the fossil record. And we can get relative growth rates of slow lamellar bone to fast woven bone. We can do the same thing with vascular canals. So again, these are the tubes in the bone that are bringing oxygen and nutrients into the bone, and they get organized or oriented in different ways. We have longitudinal canals. So these are running parallel to the long axis of a bone, like an arm bone or a leg bone. These are typically considered the slowest or associated with the slowest growing bone. Versus down here, we have radial canals, and these are going to be radiating and being perpendicular to the long axis of the bone. And these are associated with fastest growth, especially what we see in babies and juveniles as their bones are growing really fast. And these are things that we can actually measure in modern animals, and then again, apply this to the fossil record to ground truth these relative growth rates. Another thing we can do is we can, in a lot of bones, we can also treat them kind of like we do tree rings with dendrochronology and age dating tree rings. So this is one of my favorite pictures of a bone that I've cut. And I was doing, I designed a little exhibit for my museum back in Kansas that was really showcasing photos from my research, but showcasing them as art. So it was actually an art exhibit and installation. And I was doing a little promo, kind of one of our quarterly art walks we had in our historic downtown area. And I took 6 pieces that were going to be in this installation just to kind of drive, engage with the community. And I forgot all of my labels for what I took. So I had to talk to everybody that came in. And so I started asking them, what do you think this is? Because they had no clue because I forgot all my labels. And so everybody who saw this one thought it was cork or some sort of wood, because that's actually exactly what it looks like. And you see these bands and here you think, oh, these are like tree rings. Well, they kind of are. So this is actually, I'll show this one later. This is a piece of sea turtle, upper leg bone, femur bone, fossil sea turtle. And these are in fact growth rates. So in some cases, yes, we can say fossil A was growing faster than fossil B, but we can also say that this animal was 4 years old when it died. And it grew to be this big in 4 years. And that's something we can actually only do by cutting open bones and looking at these growth rates. Otherwise, we have no clue how many years old an animal was, a fossil animal was. So we can actually go in. These are called lines of arrested growth or growth marks, and we can go in and count them. So we can get a huge amount of information from just looking inside bone. And this has definitely been a growing field in paleontology over the last 30 years, I'd say, and getting applied more and more to different questions and different types of animals. And then so when before I started doing sea turtle research, I would always say that the only people who actually study osteohistology is paleontologists. It's not something that biologists typically study because they can actually go out and observe an animal, like pick it up and put it on a scale, watch it hatch, watch it die, know how old it was. Things that we can't do with fossils. And so typically, even with modern animals, it's been mostly paleontologists doing this sort of work. And then I started reading sea turtle literature and they proved me a liar because there's a huge amount of sea turtle conservation work that is done through osteohistology because you can cut open the bones and count their rings. So sea turtles, we have 7 species alive today. They're all protected because they're all threatened. And so we're not allowed to kill them on purpose, which is really good because we kill more than enough of them, not on purpose. And so how conservation biologists get their population data is by looking at dead sea turtles that wash up and get stranded, cutting them open and actually getting age profiles and relating the age of the animal through counting their rings to the size of the animal or the shell. So there's been a huge amount of research on almost all 7 of our living sea turtles that have actually looked at cutting open the bone and how these animals are growing to some degree. Again, most of it is counting up rings. So here we have a couple of examples. So we have the green sea turtle on the left, a loggerhead sea turtle in the middle, and then our Kemp's Ridley, our most endangered, on the right. And you can see that one interesting thing about sea turtle bone is that it's fairly spongy, meaning there's a lot of open space in the middle compared to most reptiles, which have very, very solid kind of dense bones. And then this one's actually a really young, actually still growing very fast when it died. And one of the things that we've seen out of all of this modern sea turtle osteohistology research is that we actually see two different growth patterns within our modern sea turtles that actually relate to the two different kind of evolutionary branches of sea turtle tree that we have. So we have our hard-shelled sea turtles, which is our greens and our loggerheads and our flatbacks, everything but the leatherback. So 6 of our species are hard-shelled sea turtles. And then we have our leatherback sea turtle. So our hard-shelled sea turtles all have bones that look pretty similar. Okay, pretty similar to this one over here, where yes, you do have these big kind of open areas, but it's pretty dense bone still. Versus this over here, this is a leatherback sea turtle. And it's just really thin bone, super spongy in the middle. And so a very, very different growth strategy and bone growth strategy within the leatherbacks versus the rest of the sea turtles that we have. And so I took this information back to fossil sea turtles. And before this study came out in 2003, there was literally not a single paper looking at fossil sea turtle bone growth, despite everything that we've done with modern sea turtle. And that kind of surprised me. So I did that stuff. Okay, so let's get back to Protostega. So I wanted to know how Protostega grew throughout its life and what that meant for its ecology and how it lived its life. So I looked specifically at fossils that came from Western Kansas. So I had access to those through my museums. I contacted other museums as well to borrow their specimens, which is always a little iffy since I am asking to permanently alter fossils, which not everybody's a fan of. But also there's actually a huge amount of data on modern sea turtles that show that populations of the same species grow differently in the different parts of the world. So like Northern Atlantic greens grow different than Hawaiian greens and things like that. So I wanted to try to kind of control as many factors as I could. And since I wanted to know how these animals grew throughout their life, I tried to put together what we call an ontogenetic series, where I could sample individuals at different stages of their life. So I tried to get some small ones and I tried to get some big ones to see what the differences were. I was able to get permission to put together a sample set of 4. This doesn't seem like a lot, like anything greater than 1 is really good in vertebrate paleontology, so I'm kind of ahead of the curve. And how I ended up between what I could use from my museum and what I was able to borrow, I pretty much had 2 small ones and then 2 big ones. So let's, and yeah, so just for reference here, the largest sample is fractured, but half of that humerus, these are all upper arm bones, these are humeri. So half of this humerus is like the length of my humerus. So these are big sea turtles. These are the ones that get to be like 10 plus feet long. Okay, so let's start with the small ones. So this is actually the one that I showed before that looked like pork. That was just a femur, and this is a humerus and upper arm bone. So you can see that it's really, really spongy, but you can also start to see some of these lines that we're talking about in them. So if we take a closer look, and this is just kind of different lighting, this is just plain light coming through down here. This is just a fancy type of light where I kind of organize it to come into 2 planes at 90-degree angles versus 360 degrees. And then I put a filter on it that turns everything magenta and it looks really pretty. Same slide, same picture, telling me slightly different things, but you can then actually see these 4 different growth lines. So this individual was 4 when it died. And you can see between years, so between age 2 and 3, like it has a significant amount of bone growth. So this individual is growing really, really fast, especially when we consider it's a turtle. And turtles are not known for growing very fast. So then I can look at one of my bigger ones. So I go to this one, this was from University of Kansas's collection. And the biggest one in my sample set, and this is crushed in the middle, which is why this kind of looks ground up, because it is, everything's kind of crushed from all the rocks that sit on top of it. But especially moving out here, you can see that again, it's this really, really spongy bone, lots of light coming through, where we have all of this open space. And I can go in and look at this again up close. And I can see here 6 different lines that I can count going through. This isn't the whole story though. So one of the things we always have to take into account is what's going on in the middle. Are there lines, are there these growth lines, so these years essentially of an animal's life that we're missing, either because it's crushed or because one of the things when we grow bone, we put bone on the outside. That's how we lay down new bone. So as we grow our bone outward, we actually absorb the old bone from the inside. So we're kind of constantly recycling our bone tissue that way. And so we also want to know, did this turtle maybe resorb any bone in here? So we're missing some of those lines. So I did what's called retro calculation, and it's just very basic. I didn't use any math at all. It was great. But I just took the smallest bone in my sample and scaled it to the same size as the largest, and then inlay them, so that the point of kind of initial bone growth is the same. And so then I can go back, and what I found was I was actually missing maybe 2 growth lines. So I had 6 preserved in kind of this orangish one in the back, and then I can add 2 more that are probably missing just from crunching or resorbing. So I put together my sample set, and the smallest one in my sample set had 2 growth marks, so it was at least 2 when it died, and then my largest ones had 8 growth marks. So how I interpret as was at age 8 when it died. And so what we're seeing a couple of things here. First, these animals could double in size between ages 4 and 8. That's really fast growth. And if I extrapolate these data to the largest specimen that's ever been found of this type of sea turtle, that these things are possibly reaching full body size, so 10 to 12 feet long, within 10 years, which is fast, like even for a mammal, much less a sea turtle. So we're seeing really, really fast growth rates with this. And if we look at the general pattern of what Protostega is doing versus our modern sea turtles today, we see bone growth patterns, both the rate of bone growth as well as these patterns of spongy bone, high vascularity, high growth is much more similar to leatherbacks than it is to any of our hard-shelled sea turtles, which is really, really cool because leatherbacks are super cool. Like they are just the weirdest animals that I've ever come across. They do all sorts of things that sea turtles and reptiles traditionally thinking shouldn't do. They have a large body size, so they can get to be, 2.5 meters, so they'll get to be 6 to 8 feet, 6 to 8 feet long. They show really rapid growth rates compared to hard-shelled sea turtles. We don't actually know how fast they're growing, but even though they're like twice as big as a green or a loggerhead, they're also growing twice as fast and reaching adulthood probably twice as fast. They do weird things like migrate to the Arctic and go to really cold waters, so places where you shouldn't see something that's cold-blooded or ectothermic. They dive down to like 1,000 meters. I don't know why. I mean, they're eating something that has to have like the nutritional value of a plastic bag because all they eat are jellyfish. But they're diving down there and seem to enjoy it. And then the coolest thing is that they actually hold body temperatures higher than their surrounding water, which is really weird for any sort of traditional reptile alive today, other than birds. So there's a study in 1990 that looked at this. And so this highlighted in blue area right here, these lines are showing reptiles on top and green sea turtles on the bottom, those parallel lines of their resting metabolic rates. So if they're just hanging out on a beach, what their resting metabolic rates are. Here in pink, that's what leatherbacks do. So they have actually a resting metabolic rate that could be scaled to something like a mammal, which is this line up here. And so we have resting metabolic rates that are 3 times higher than what would be predicted for a typical reptile of their size. And so in this paper, they actually coined the term gigantothermic to describe leatherback sea turtle metabolism. And then this has been taken into the dinosaur world of, yay, dinosaurs are gigantothermic. It's not that straightforward because baby leatherback sea turtles have really high metabolic rates and they don't stop moving. They don't have a rest period. They just swim constantly. And so their babies, before they get really big, still have these elevated metabolisms. So something weird's definitely going on with that, which is really cool to think about. And so similarities in bone growth patterns between the two can be used to infer behavior. That if you have similar growth, then you could infer similar life history patterns between our extinct protostega and our living leatherbacks, which would include rapid growth to reach large body size quickly, and also rapid growth to reach sexual maturity quickly. One of the cool things about sea turtles is that when they reach sexual maturity, they like cut off growth and they're just like, all energy is going to sex and babies, no more energy to growing big. So when you find a sexually mature sea turtle, that's as big as it's ever going to get. So if we find a sea turtle that stopped growing, we can make inferences that it was sexually mature at that point in time that it died in that age. So really, really cool things that we can correlate between the two just using the skeleton. Other things like sustained swimming. And then I also think that protostega could have had this elevated body temperature similar to how leatherbacks do today. So this isn't the whole story though, because I also cut up other sea turtles. If you're going to cut up one, you might as well cut up others too, especially when you're in charge of a museum and I can give myself permission to cut things. It was a pretty sweet deal for cutting up sea turtles. So this is called a phylogeny. So this is showing the evolutionary history of different species of sea turtles specifically. So up here, Dermocheles, this is our living leatherback. And then this blue group right here, these are our hard-shelled sea turtles. In this predictive model, green, these are our protostegids. So down here we see protostega. So this is protostega, what I've been studying, and then its closest family members on the sea turtle family tree. And so I cut protostega, but I also cut Desmatocheles. Specifically, I cut Desmatocheles lowi. So it is a sea turtle that lived a little bit earlier in time. So it's a little bit older, lived a little bit earlier in time than protostega, and is deeper down the family tree. So this is like a couple of steps removed if you think about your family history and your family tree. So they're more like, like 7 or a couple of cousins removed versus brother sister. So I cut that one and then I compared it to what's going on with protostega. So this is Desmatocheles. So this is still a decent sized sea turtle. This is not a little sea turtle. This is something that's going to be at least as big as our leatherbacks today. So still a very large sea turtle. So you can see half of its skeleton compared to a human. So this is at least an 8-foot, probably sea turtle. And I cut its bone opens, or its humerus, so its upper arm bone. That's what's being shown here. And then I cut it right in the middle where I cut all the other ones right below this little projection. And this is what the bone looks like. So this bone, what I have just highlighted here in these hot pink arrows are all of those growth lines. So you can see that the growth lines are much closer together and that bone between those growth lines is a lot denser. It's not as much of that spongy until you get into the middle, which is where it was broken. And so with this, I have Desmatocheles, this kind of more what we call more basal or more primitive, for lack of a better word, sea turtle within the same family as Protostega, growing like a hard-shelled sea turtle, having sustained longer but slower growth, versus our Protostega, which was growing very, very quickly. So this one here, I think I counted 15 growth lines in, and it was still growing, versus the 8 to 10 that we have in our full-sized, much larger Protostega. So my question was, what happened? So these are related animals in the same evolutionary lineage. And so somewhere between Protostega's evolution, or sorry, somewhere between Desmatocheles' evolution and Protostega's evolution, we see this switch to really, really fast bone growth. So I wanted to know what happened. I'm an ecologist. I want to know if there's something going on in the ecosystem that could be spurring this along. So looking at kind of the late Cretaceous or the Cretaceous and the evolutionary history of these animals, we see Desmatocheles as a genus showing up first about 120 million years ago. Then we see the evolution of mosasaurs. So mosasaurs are one of these big marine reptiles. They are the biggest apex predators of the ecosystems of this time, made famous by things like they finally made an appearance in Jurassic World. So this is the Jurassic World. This is way bigger than mosasaurs got, but it made for a fantastic scene, and it was good to see something other than dinosaurs. So we saw mosasaurs or apex predators show up about 100 million years ago. After that, we see another Desmatocheles species, lowi. Those are the ones that I cut open, that we also get from Kansas. And then about 85 million years ago, we get Protostega. So what happened between Desmatocheles lowi and Protostega gigas? We get Tylosaurus. So Tylosaurus is pretty much the big boy of mosasaurs in these late Cretaceous ecosystems. This is one of the biggest mosasaurs to ever live. Tylosaurus prorigor, we know they lived side by side with these big Protostega sea turtles. We had smaller species that lived earlier with things like Desmatocheles. So one of the big things that we see changing in these ecosystems is the evolution of really large predators. And so I think that is really what drove kind of this evolution and the change of how these sea turtles were growing. Because growing large is a great defense. So large size is a great defense against getting eaten. You know, the largest animals that we have on Earth are all herbivores and are much larger than their predators. Elephants, for example, are one of the very few animals other than humans that actually grow to old age and die of old age because they outgrow all of their predators. So growing fast to a large size is a great survival strategy. And since in sea turtles, adult body size is correlated with sexual maturity, they're not only growing fast, growing big faster, they're actually reaching sexual maturity faster, which is also a great survival strategy because you can start having babies sooner and potentially more babies through your life. So all great survival strategies that we have. So in conclusion from this study, Protostega had bone microstructure very similar, so growth rates very similar to what we see in leatherbacks, more so than what we see in our hard-shelled sea turtles. And Protostega, these growth patterns reflect rapid growth to a very large body size, meaning that they're also reaching reproductive timing sooner as well. And that we also then have the evolution of this change to the larger body size and the faster growth rates is potentially correlated with the changing ecology of the time. So the introduction of new apex predators to these unique marine ecosystems. What's unclear left, or what's left unclear, is whether this elevated growth rate and this change in metabolism is something that happened once in sea turtle evolutionary history or has had multiple times that it's evolved. And that's because if you know one thing about sea turtles or about turtles in general is that nobody understands turtles. Like they make no sense. They show up and kind of pop up in the fossil record, being like, hey, we're turtles and we look like turtles and we're always gonna look like turtles. They don't change that much, and we really have a hard time figuring out like where they came from and how they're related to each other. And they also have their shoulders inside their rib cage, which is really, really weird. They also have their hips inside their rib cage, which is maybe even weirder. But so there are a couple of different things. So I showed this evolutionary tree before, where we have our protostegids down here as what we call the sister group, so share a common ancestor with our leatherbacks. So there are interesting things here. So we know that we have fast bone growth here, and we know that we have fast bone growth up in our leatherbacks, and we know that we have slow bone growth here. So this would show us that potentially within this evolutionary hypothesis that we have two different times that fast bone growth has evolved. So what we would call convergent evolution. So they're converging on the same growth patterns. There have been other hypotheses though. So this one over here is actually showing our leatherbacks coming out of the protostega group. And there are a couple of different sets of researchers that have hypothesized this evolutionary relationship, that leatherbacks are just survivors of the same protostega lineage versus a different offshoot. And so this would actually, you could conclude that maybe this pattern only evolved once. And it was the survivors that made it past the big end Cretaceous extinction 66 million years ago that had the advanced or the faster growth rate. So there's still a lot of questions. There are ways of testing this to some degree, but it's really going to come down to people who do a different type of science that I do to figure out these evolutionary relationships. But could be a cool piece of data to inform some of these evolutionary relationships. So like any science project, you always come out of it with more questions unanswered than you go into it. So there's a lot of cool stuff, and a lot of people who helped me get access to turtles to cut up. And again, I'm very grateful to this project because it really kind of spurred me into where I am now, starting to think about more than just past ecosystems and the implications of them. So that's all I got, and I'm happy to answer any questions.
All right, thank you very much, Dr. Wilson. Thank you for grabbing.
Question?
Hold on just a second. So for our, I missed it. I apologize. I'm not our normal Zoom person, so I'm like a little bit behind the scale today. But for our in-person people, we you should have grabbed a note card from the back table. If not, I can grab them around, bring them around. We have you write down your questions so that I can read them for the Zoom followers as well. If you are on Zoom, please put any questions in the Q&A, and I will read those to Dr. Wilson. But you want to just
Yeah. Now, your Western Kansas is where you do your work.
Yeah.
And okay, what's the topography like, and how deep do you have to go? Or are they exposed?
It's all exposed. So fossil hunting is extremely low tech. You literally just walk around and look at the ground. And something has to be physically weathering out, falling out of a hill to be able to find it.
Yeah.
Western Kansas is flat. You know, Kansas is flatter than a pancake. That definitely comes to life when you drive across that part. There is some topography, so pretty much cut out from draining glaciers, you know, 100,000 years ago, that have cut out some channels where we have rock exposed. But maybe, I don't think I've ever seen outcrops more than in the extreme, maybe like 40 to 50 feet high. Usually it's maybe like 10 to 12. And sometimes it's just flat and a farmer hits something with a plow. You know, I've gone out, and it's a different age, but I've gone out, I look at a bunch of like mammoth tusks that get hit by road graders, and that's how they were discovered.
When was the old age? I guess of the rock. The deepest, like, 40 feet.
And so it's going to depend on where you are in the state. So in Western Kansas, for the most part, maybe like 92 million is about as as old as we go, but you can go to the eastern part of the state and they actually have stuff from the time period before the dinosaurs, the Paleozoic, that goes back to maybe like 320 million years old. There are like some of the first amphibians that come from that area, and then a lot of sea creatures like brachiopods and clams and trilobites and stuff. So it depends on where you go and what rocks are exposed within the state. So same here. So, I mean, a lot of the shark's teeth that we get that are washing up on our Atlantic coastline are going to be Miocene in age, so 5 to 10 million years old. But there are a lot of places kind of in north central Florida where you can find those ice age, like the mammoths and the mastodons and the giant ground sloths and the giant birds. So it just depends on where you go.
Thank you.
We have a question. Do you still have access to the equipment to cut your sea turtle bones?
I don't. So that was one of the decisions when I made when I changed careers, is I did leave my lab behind. And I knew that was going to be a part of it. So there are people at, I think it's University of Central Florida, that there are sea turtle researchers that don't do the same type of research that I do. So there might be ways of collaborating, but I'm a little bit more interested these days in how to use the data that we have from past marine ecosystems to look at kind of longer term solutions for our modern ecosystems, or better predictions for our modern ecosystems. So that was a very conscious decision that I made when I left the academic lifestyle. And so far I've been happy with my decision making. But I left a lot of questions for other people. One of the things that I did, God, I felt so bad about it. So 2021, there was the polar front that came through in like February and hit, the polar vortex, and it hit Texas really hard. And so Texas had tens of thousands of sea turtles that were cold stunned. So when seawater, or when the air temperature and seawater get too cold, sea turtles, hard-shelled sea turtles, because they're cold-blooded, their system stops working. They get too cold to move, and so they essentially drown. And so cold stuns, they were pulling them out of the Gulf of Mexico, trying to save them. And I felt so bad. People knew I was working on sea turtles and sent me all these articles about like dying sea turtles. And so I'm contacting these people and I'm like, you're doing God's work. Can I have your dead ones? And essentially it's like, not all the sea turtles can live, right? And it's a lot easier, like you can't get permits to kill sea turtles, which is good. I'm not advocating for that. So you work with already dead sea turtles. So I asked them if I could have any of their dead sea turtles. And sea turtles are really interesting. If you want to study sea turtles while they're in the water, you have to go through NOAA for permission, the National Oceanographic and Atmospheric Administration. And if you want to work on sea turtles while they're on land, you have to go through Fish and Wildlife. So like there are all of these different permitting avenues that you have to go through. So I ended up piggybacking on a place in Texas, Sea Turtles Inc. Permits. But by that point, they'd already gotten rid of like all their dead sea turtles because they're huge and they're stinky. But they were actually able to collect a size gradient. So I actually have sitting in Kansas, if I ever do want to work on them, I have a baby, a medium large, and large sea turtle flippers to work on to do this same study on modern green sea turtles. Because even though there's been all of this osteohistology work on modern sea turtles, it's all just like cut it up, see how old it was and how that relates to body size and where it is and things like that. Nobody's actually looking at these kind of growth rates that change through the life of the animal. So it's a project that's out there for some.
I have
Okay.
a few questions I will get to you, sir. Based on what you have found in your research, are you starting to form other hypotheses on modern sea turtles and the ecosystems in which they live?
So I think yes. I mean, the easy answer is yes. I have so many questions. One of the first ones, which is when I really started getting in touch and talking to sea turtle conservation biologists, is because we hear everything obviously about warming temperatures. Sea turtles are reptiles, so if you think about crocodiles and things like, and alligators, their eggs, just like crocodiles and alligators, the sex of the embryo is determined by the incubation temperature. So that's one of the big concerns now with warming climate, is that we're hatching more females than males. And so there's this big conservation protection to protect those late nests. So the sea turtles that are laying in like August and September, because they'll be incubating in cooler sand temperatures, thinking that those will hatch more males. But I'm dealing with in the Lake Cretaceous, I'm dealing with a greenhouse climate. I mean, we have temperatures that in the Arctic are 15 degrees Celsius higher than what we see today. So it's extremely hotter as you move to higher latitudes. And sea turtles thrive. So we have at least one new species that needs to be described, maybe a second one, which puts us at 8 or 9 species of sea turtles just within this like Kansas section of the Western Interior Seaway. That's huge sea turtle diversity. We only have 7 alive today in all of our global oceans. So obviously these sea turtles are doing very well. So I think there's a lot, I don't know how to answer the questions, but I have a lot of questions about what sea turtles were doing in the Lake Cretaceous when it was really hot year round and still having these healthy populations. I do think there are other things that are really interesting when we look at survivability across ecological disturbances. And I think that's where we can get a lot of information to move forward with, is we have sea turtles surviving. Which ones are surviving and what's unique about those populations? There's some things that are just hit or miss, right? Like big space rock hits Earth 66 million years ago. If you get hit by the space rock, you die. Like there's just some things that are bad luck. But there are other shifts that we see with kind of smaller changes in climate, changes in sea level that was going on at the time, to look at how those all trace and track with survivability and diversity of different animals. And I think there's a lot to learn through that. And we've looked at like big mass extinctions, but we haven't looked at those smaller climate permeations as much.
Question relates to the age.
Uh-huh.
In your example for the statement, 8 years was the oldest that you
That was the oldest that I cut, was 8 years old. Mm-hmm.
seen any other indications of anything much older? Thinking modern sea turtles don't reach sexual maturity until 20-some years.
Right. So we really don't understand sexual maturity in our extant, our living sea turtles, very well. We definitely understand it better in the hard-shelled sea turtles, especially the greens and the loggerheads. And those estimates are 35 to 50 to reach sexual maturity in greens and loggerheads. Leatherbacks, it's shocking how little we know about leatherbacks. And there is not consensus on what age of sexual maturity is for leatherbacks. Some people do put it at 25 based on mathematical calculations, but some people actually have it at 5 to 7 years old. Which, looking at their bone growth, I'm actually on that 5 to 7 scale. So I think they're growing really, really fast in a short amount of time, which could kind of be on par with what we're seeing in the protostegas as well. But there's a lot more modern sea turtle studies that need to be done, especially on the leatherbacks. There's been like one guy that studied leatherbacks, and I had his slides. That's where that pink one came from. He sent me his slides when I was starting this study. So there's a lot more. We don't have a good growth series, and leatherbacks. So things are changing and getting better in terms of verifying data now that our satellite tagging has improved. So now we're tagging animals, and then when they get stranded and they died and they wash up, we can actually figure out how old those animals and track data a lot better. So I think that's going to help. I just don't know how many people are doing that with leatherbacks, because leatherbacks they spend their entire lives at sea, which is really unique for sea turtles. They don't have any sort of near shore, we're going to hang out in the lagoon eating seagrass like greens stage in their life. They hatch, they just take off, they literally never stop swimming. And then like, they're like, hey, we're going to go up to, Nova Scotia, and then we're going to go over to England, and then we're gonna go down to the west coast of Africa, and then we're gonna come back to Florida or something like that. Like they go on these huge migrations. And so they're really, really hard to track. So there's a lot of research that needs to be done. So great questions. I don't know. I don't think anybody has the answers to them right now.
We have one Zoom question.
Uh-huh.
What do you think has been the most important thing you've learned about past sea turtles for the conservation of present and future sea turtles?
So I think the most important things haven't been answered yet. So what this study ended up doing was setting the baseline. This was really the first study to look at what fossil sea turtles and ecosystems, but we have a pretty good sense of at least all the players in the ecosystems. There's a lot of people that are looking at what sea temperatures were doing, what oxygen content was doing, and things like that. So really, I think the most important thing was actually having that first study that sets a baseline that we can now catapult other studies, build the sample set, add other turtles, add other ecosystems. We have a record of fossil leatherbacks, work with some of them. They're post dinosaur extinction, but definitely went through warm periods, cold periods in the last 50 million years of Earth's history. So there's a lot of studies that could come out to kind of piggyback on this one and springboard off of this one.
Any further questions from in person? You answered all our online
Okay.
questions.
Excellent.
All right, thank you guys
Well,
all
thank
for
you.
coming. We appreciate it. And thank you, Dr.
Yeah.
Wilson.